We had earlier learnt about the basic concepts in biotechnology. We understood that we have a modern definition of biotechnology. Biotechnology is primarily a science, interdisciplinary science, which integrates genetic engineering or recombinant DNA and its sort of methods along with plant tissue culture. In plant biotechnology, we understand we need to develop the first type of recombinant DNA. Recombinant DNA is the sort of an answer for many of the problems of the 21st century. Applications of biotechnology run into agriculture, medicine, forensic science and environment. In today's chapter, we will be reading about the tools in recombinant DNA technology. The meaning of the term tools is what chemicals or what physical and chemical methods would you use to cleave the DNA or break the DNA into smaller pieces. How after you isolate the sort of a DNA from a plant cell. From a plant cell, we understand the DNA is present in the chromosomes and chromosomes are present in the nucleus. So we need to break down the cells, get into the nucleus, break down the chromosomes, and we need to biochemically characterize the sort of a DNA. We need to isolate the DNA. After we isolate the DNA is a long stretch of nucleic acid made up of phosphodiester bonds and made up of basic units which are termed as nitrogen bases. And the nitrogen bases are adenine, guanine, cytosine and thymine. We use in biotechnology, initially we use a basic tool which is termed as restriction enzymes. Now, what exactly are restriction enzymes? The topic of the day is restriction enzymes. Restriction enzymes are normal enzymes which are inherently present in the bacteria. All bacterial cells, especially Escherichia coli, have native resident restriction enzymes naturally present within them. Thereby, what is the purpose of the restriction enzymes? And we'll have to be dealing in the chapter of restriction enzymes. We'll exactly be looking into issues such as what is meant by a restriction enzyme, what happens when a virus infects a bacterial cell. We'll also understand why they are termed as restriction endonucleases, because they are present within the sort of a cell. Most importantly, we'll be studying two important type of restriction enzymes. One is termed as HIND2 and another one is termed as ECOR1. We'll also be addressing towards the nomenclature or the way how restriction enzymes are named. We understand that restriction enzymes primarily are specific enzymes which are used as tools in molecular biology to break the sort of a DNA sequence. So when they break the DNA sequence or when they cleave the DNA sequence, they are termed as molecular knives or molecular scissors. And the restriction enzymes can cut the DNA at specific sequences. That is, the restriction enzymes recognize only certain particular sequences in the DNA. We'll also be studying about what is the recognition sequence, why does it recognize that particular specific sequence, and how does it cut. So when we take the sort of a DNA, in the DNA we have certain sequences which are similar in identity on both the strands. If you take the nitrogen bases, and the nitrogen bases which moves from the left to right on one strand and from right to left on the other strand, both of them are having the same sort of a sequence. Such sequences which are present on the DNA are termed as palindromic sequences. An easy example to remember palindromic sequences is by means of analogy. We use two words called Malayalam as well as Liral. When read on either side from the left to the right or from the right to the left, they spell the same. So such sort of a sequences would be recognized by restriction enzymes. So in the study of restriction enzymes, we will also be knowing about A, recognition sequence, what are palindromic sequences. And when a restriction enzyme cuts the DNA, what is the method of cutting? It can just cut like how a carpenter cuts a wood straight, like a blunt end, or else a carpenter can shear a particular sort of a wood and cut it by means of a staggered end. So the cutting pattern is a staggered cut or else a blunt cut. So we'll also be understanding the mechanism of how restriction enzymes cuts the sort of a DNA. And at the end of the chapter of restriction enzymes, you will also be knowing about their applications in molecular biology. So, we start off with relation to this chapter of restriction enzymes. Restriction enzymes are the first sort of tools in recombinant DNA technology. Along with restriction enzymes, we also have, as part 2, we also have to be learning about what are the cloning vectors, or which are the sort of molecules which have to be loaded from one bacteria to another bacteria which carry the foreign gene. These are termed as vectors. So, after a vector or after a sort of a virus or a DNA moves from the host to a recipient cell in which it has got a genetically altered gene present in the sort of a DNA, that gene should be able to be cultured in the sort of host cell. After getting into the host cell, we need to look at issues such as what is the host, how do you say that the host is competent, that is, it is capable of taking a sort of a gene. So, definitely in the tools of recombinant DNA technology, we will be studying as part A restriction enzymes, part B we will be studying about cloning vectors, part C we will be studying about competent host, part 4 we will be studying about transformation of the host cells when a foreign gene is inserted, and later on we will exactly be studying like what do you do with a particular sort of a gene. So, all these particular specific sort of steps are primarily termed as tools. So, after learning about the tools, then in another block you will also be learning about the sort of processes in recombinant DNA technology. That is, what are the methods by means of which you could isolate a DNA, isolate a gene of interest, amplify a gene of interest, and later on look in terms of how do you make multiple copies of the sort of gene of interest as well as get the sort of a product for human use. So, we start off with relation to restriction enzymes, which are the first tools in recombinant DNA technology. When we take in Escherichia coli or when we study Escherichia coli, we understand in Escherichia coli there are two very important type of enzymes which are natively present within the sort of a bacteria. And these enzymes, which are termed as restriction enzymes, can break down or slow down or inhibit the growth of a bacteriophage. A bacteriophage is a virus which can infect a bacteria. And when you take a bacteriophage, the bacteriophage can send its sort of a DNA into the bacteria. So, during infection, when the bacteriophage releases the naked DNA into the bacteria, there is a high probability that the bacteria will get killed, or it will primarily lead to death of the bacteria. To avoid the death of the bacteria or to fight out the sort of an infection, bacteria inherently enzymes which are termed as restriction enzymes. So, what do the restriction enzymes do? The restriction enzymes will prevent the foreign viral DNA to enter into the sort of a bacteria, and it would cleave the sort of a viral DNA. And because it cleaves the viral DNA and restricts the progression of the viral DNA into the bacterial cell, they are termed as restriction enzymes. When you take restriction enzymes in the case of bacteria, we have two different types of enzymes. We have one type of an enzymes which adds methyl groups to the host DNA, and we have other sort of an enzymes which cut the DNA of the invading virus. The enzymes which cut the invading DNA in the host cell, they are termed as restriction enzymes. If we look at restriction enzymes, they cut the foreign DNA, and that's why they restrict the movement of the foreign DNA. And because it occurs within the sort of a bacteria, that's the reason why they are termed as endonucleases. Now, when you take the restriction enzymes, the first of the restriction enzymes which ever has been isolated and characterized comes from a sort of an enzyme which is termed as HindII. HindII is the first restriction enzyme which has been discovered in the world, and HindII is a restriction enzyme which has been first isolated and characterized. It has been isolated and characterized from E. coli RY13. To date, more than 900 restriction enzymes have been isolated from over 200 strains of bacteria. So, we have a wide array of restriction enzymes which have been isolated from more than 230 strains of Escherichia coli itself. And HindII always cuts the sort of a DNA, cuts the viral DNA by recognizing a specific sequence which is termed as the restriction sequence. That is, HindII is a restriction endonucleases which will scan the sort of a DNA of the virus. It recognizes a particular nucleotide sequence, and the particular nucleotide sequence is recognized and it is termed as a sort of a recognition sequence. And normally when you take restriction enzymes can recognize 5 base sequences, 6 base sequences, 4 base sequences. And how do you primarily know about the restriction enzymes is there is a particular pattern of nomenclature of restriction enzymes. Next, we get into the nomenclature of restriction enzymes. We are now dealing with relation to nomenclature of restriction enzymes. Whenever we want to name a restriction enzyme, which is a sort of a tool in molecular biology, restriction enzymes are always given the name according to a particular sort of a pattern. Like if you take the restriction enzyme called ECOR1, what is exactly the meaning of ECOR1? E refers to Escherichia, CO refers to coli, R refers to the strain, and Roman 1 indicates the strain or the order in which it is isolated. If in a lab we have the second strain and the third strain of ECO getting discovered, they are termed as ECO R1, R2 and R3 depending upon the series or depending upon the time of the discovery. Now similarly, if you take the second example, if you take HindII, HindII is a restriction enzyme which has been derived from Haemophilus influenzae. Haemophilus influenzae is the name of a bacteria. Haemophilus influenzae has got a strain called RD taken as D. So thereby the name is given as HIND. And the first order of discovery, the first restriction enzyme which has been discovered from Haemophilus influenzae is termed as HIND1. Then the second order is termed as the second, is termed as HindII, and later on you got HindIII. Thereby, in any particular nomenclature of a restriction enzyme, we have the name of the genus, we have the name of the species, we have the name of the strain, and again we have the order of the discovery as seen or as exemplified in ECOR1 and in the case of HindII. ECOR1 is always discovered from Escherichia coli and HindII has been discovered from Haemophilia influenzae of the sort of strain D.